Hello friends, we have already discussed prokaryotic DNA replication in detail. We have also seen overview of eukaryotic DNA replication. You can refer my videos, the link is given in the description box. Today we are going to discuss about enzymes and proteins involved in prokaryotic and eukaryotic DNA replication. So let us see them one by one. First of all, origin of replication both in eukaryotes and prokaryotes is recognized by initiator protein. In case of eukaryotes, it is origin recognition complex. In prokaryotes, it is DNA protein. Then the unwinding of double helix of DNA takes place. It involves helicase enzyme. This helicase enzyme separates the two strands of DNA. In case of the eukaryotes, it is MCM27, which is hexamer. And this helicase loading is directed by ORC, CDC6 and CDT1. This we will discuss later in initiation step of eukaryotic DNA replication. And helicase enzyme in prokaryotes is DNA-B. This DNA-B is loaded by DNA-C. Hence, it is known as the helicase loader. As the two strands of DNA are separated, it is important to stabilize the single strands of DNA in order to prevent the rewinding or reannealing of the strands. In eukaryotes, it is stabilized by replication protein A or RPA. In prokaryotes, it is single strand binding protein. Two replication forks are formed at each origin, and these replication forks move away from origin in opposite direction to each other. As these proceeds, it results in DNA supercoiling both ahead and behind the replication bubble. This also we have already discussed. You can refer it. This supercoiling prevents the further movement of replication fork. Hence, it is relieved by topoisomerase 2 enzyme both in eukaryotes and in prokaryotes. Now the priming takes place, that is the synthesis of RNA primer. Because DNA polymerase cannot initiate the synthesis of new strand. It requires 3-OH group which is provided by RNA primer. And this RNA primer is synthesized by primase enzyme. In eukaryotes, RNA primer is synthesized by DNA polymerase alpha enzyme. So DNA polymerase alpha has 4 subunits, 2 subunit function as the primase. In case of the prokaryotes, it is synthesized by DNA G. This DNA G or the primase is activated by DNA B, that is helicase enzyme. Now the extension or the synthesis of the new strand of the DNA takes place. This takes place by the main replicative enzyme. In case of eukaryotes, it is DNA polymerase delta and DNA polymerase epsilon. If one strand is synthesized by DNA polymerase delta, then the other strand is synthesized by the DNA polymerase epsilon. pCNA, proliferating cell nuclear antigen, is the clamp which holds DNA polymerase in place when the nucleotide is added or when the extension of the new strand of the DNA takes place. And RFC, replication factor C, is the clamp loader. In case of the prokaryotes, DNA polymerase 3 is the main replicating enzyme, which is the holo enzyme, has 3 parts. One is the beta clamp, which holds the DNA polymerase in its place. And the clamp loader is clamp loading complex, which consists of gamma, delta, delta dash and xi subunit. After the synthesis of the new strand, RNA primer is removed and it is replaced by TNA. In case of eukaryotes, primer is removed by RNase H and FEN1 and replaced by floating enzyme DNA polymerase delta. In case of prokaryotes, RNA primer is removed and replaced by DNA polymerase 1. There is nick or gap present between the DNA chains, which is sealed or ligated by DNA ligase enzyme both in eukaryotes and in prokaryotes. Then takes place termination of DNA replication. In eukaryotes, there are multiple origin of replication and hence multiple replication bubbles are formed, and the termination of the replication takes place when the two adjacent replication bubbles merge. In case of the prokaryotes, there is single origin of replication and hence only one replication bubble is formed, and termination occurs when the two replication forks meet at the ter site. Ter complex arrests the two replication forks. In case of the eukaryotes, telomerase enzyme is involved in end replication, that is the replication of telomere, as the DNA is linear. But in prokaryotes, since the DNA is circular, so there is no need of end replication. Now let us see the similarities between prokaryotic and eukaryotic DNA replication. DNA replication occurs before cell division in prokaryotes and also in eukaryotes. Both in prokaryotes and eukaryotes, double-stranded DNA is involved and the direction of replication is 5' to 3'. That is, new strands are extended in the 5' to 3' direction, or the nucleotides are added in the 5' to 3' direction. DNA replication is bidirectional. At each origin, replication bubble is formed. Two replication forks move away from origin in opposite direction to each other. Hence, replication is bidirectional. This we have already discussed in separate video. You can refer it. So, it is semi-conservative. One strand is the parental strand and the other is the newly synthesized strand. Hence, it is half conserved. So, it is known as semi-conservative. It is semi-discontinuous. One strand is synthesized continuously. It is known as the leading strand, and the other strand is synthesized discontinuously. It is known as the lagging strand. RNA primer is required for new strand synthesis. DNA polymerase enzyme cannot initiate the new strand synthesis as it requires 3'OH group. And this 3'OH group is provided by the RNA primer. Hence, it is required for the new strand synthesis. DNA replication has 3 steps or sequences. Both in prokaryotes and eukaryotes, there are 3 steps involved in the DNA replication: initiation, elongation, and termination. This is all for today's video. In the next video, we will discuss about differences between prokaryotic and eukaryotic DNA replication. So stay tuned. If you like this video, please hit the like button, share it, and subscribe my channel. Thank you.